So in previous part, we have seen DNA as a genetic material and RNA as a genetic material. So we have seen that in most of the organisms, DNA acts as a predominant genetic material, and in viruses, bacteriophage, RNA acts as the genetic material. So now the this we know today that DNA is a genetic material and RNA is a genetic material in some of the viruses. But after the Mendelian experiment, lot of scientists they have to do a lot of research work, and after their research work and their continuous experimental work, now today we can say that DNA is a genetic material. So in this we can say in this experimental steps, the most important one is the Griffith experiment that we are going to see today. So Griffith is a scientist, biologist who performed an experiment on bacterial strain that is Streptococcus pneumoniae. So he performed an experiment with the help of Streptococcus pneumoniae, and he told about the transforming principle. He told us about the transforming principle. See, some Mendel has also done so much work with the Pisum sativum, and he told that there are certain factors which transmit from one generation to another. So Tom has limited the research to the chromosome, that there is something in the chromosome that transfers from one generation to another. Morgan also has done certain experiments with Drosophila and all that we will see in later slides. And other scientists also they have worked a lot, and they have narrowed the search for the genetic material that is to the chromosome, that is chromosome which is located in the nucleus. Now other scientists they were having a responsibility to find out what exactly act as a genetic material. So Frederick Griffith he performed an experiment with the Streptococcus pneumoniae. Now what he has done, he has taken living S strain of Streptococcus pneumoniae. Now what is S strain? S means smooth. Smooth means a bacterial strain which can produce a capsule around it. Capsule is smooth in nature, so a bacterial strain, S strain, when we say S strain Streptococcus pneumoniae, we mean that a bacterial strain which can produce capsule around it. And the bacterial strain which can produce capsule around it, it has virulence property. What it has? It has virulence. Virulence property means this bacterial strain is able to cause disease in living organism, because it is when it is having capsulated, when this bacteria is capsulated, it will be very difficult for the immune system or the WBC to identify this. So they will not be able to kill this bacteria, and this bacteria can cause the virulency in the organism. So when this living S strain bacteria was injected into the mice died, and living S strain cells were found in the heart of the mice. So the next we have taken living R strain. R is known as rough. What it is known as? Rough strain. Rough strain means this bacterial cells are not able to produce capsule around it. So they are easily identified by the immune system of mouse, and they can be easily killed. So in this case, what will happen? The mouse will not die and it will remain healthy, and we will not found any bacterial cell in the heart of the mouse. So next, what Griffith has done, he has taken living S strain. So this is the S strain. So he has taken living S strain and he heat killed them. Okay, so he has given some heat treatment to them. So he heat killed them. So this is the heat killed S strain. So this is heat killed S strain. So he has heat killed this S strain, and then it was injected into the mice. So when this was injected into the mouse was healthy. It means that this S strain was killed, and now its virulence property has gone. It has it has given his virulence property, and now no death was, mouse was not dead, and there was no bacterial cell found in the heart. Next, so this was the first, this was the second, this was the third. Now fourth, what he has done, he has taken living R strain bacteria. So you can see he has taken living R strain bacteria and he mixed them with the heat killed bacteria, heat killed S strain. So what he has done, so he has taken the living R strain bacteria, and he mixed that with the heat kill S strain bacteria, and then he injected that into the mice. So what he found? He found that mouse died and living S strain cells were found in the heart. As we know that here S strains we have taken, but this S strains are heat killed. But this living R strain, which is non-virulent, this was converted into virulent. Now, how it was converted into virulent? So, Griffith concluded that there is some message, there is some information which passed on from this S, or we can say this living S strain smooth bacteria which was virulent to this non-virulent bacteria. So there was some message or there was some information which passed on from the heat killed S strain to the living R strain, and this living R strain which was non-virulent, it was converted into virulent. So this was all about the Griffith experiments. So what Griffith has told? Griffith has told us about the transforming principle. Okay. So this is a major experiment which we must know. Okay. So in this way, still the biochemical nature of genetic material was not known. Only Griffith was able to told that, or Griffith was able to conclude that some transformation principle, that is transform from heat kill S strain and enable the R strain to synthesize a smooth polysaccharide code, and this becomes virulent. It may be due to the transfer of genetic material, but still the biochemical nature of the genetic material was not identified with the statement. Other scientists, they also perform now after this, other scientists, they have also performed certain experiment to know the nature of this transforming principle. Either it is a DNA or it is RNA, it is a protein or what it is. It's a polysaccharide or it is a lipid. What is the transforming principle which goes from one cell to another? So next was the experiment which was conducted by Avery, McLeod and McCarthy. So what they have done, they have done certain experiment on transforming principle experiment only. What they have done, Avery, McLeod and McCarthy, this three, they perform an experiment to determine the biochemical nature of transforming principle in Griffith's experiment. So they know that in Griffith experiment some transforming principle was there. So they wanted to know the biochemical nature of that transforming principle. So they perform an experiment. Now what they have done, they purified biochemicals. So you can see they purified biochemicals, that is polysaccharide, lipids. Yes, spelling is wrong, so I will just correct it. So they what they have done, they purified biochemicals, that is polysaccharide, lipid, RNA, protein and DNA from heat killed S cell. So they have taken this heat killed S cell. Okay, they have taken this heat killed S cell and they have extracted all these biochemicals. And now they wanted to check which one could transform the live R cell into S cell. So they wanted to check that which one of this biochemical can change the rough cell, that is the non-virulent, into S cell, that is the smooth cell, that is the virulent one. So with this experiment, they discovered that only DNA alone from S bacteria causes R bacteria to become transformed. Okay. So what they have done, they have added, they have taken this polysaccharide and then they have added enzymes. In this tube, what they have done, they have added the enzyme which destroyed the polysaccharide. Then they injected it into the mice, so the mice died. Then they have taken lipid digesting enzyme in this test tube, injected into the mice, mice died. Then they have taken RNA destroying enzyme, that is RNase, and then they injected into the mice, still the mice died. Means what does means? See, in this complete thing we can see that polysaccharide is not responsible for virulency. Lipid is not responsible for virulency. RNA is not responsible for virulency. Here again in this tube we have taken the enzyme which destroys the protein, that is proteinase. Again injected into the mice, mice died. It means that virulency is again not affected by protein also. Then what we have done? We have taken DNA. We have taken a DNA digesting enzyme. Okay, so what we have done, we have added this into, we have taken DNAs, that is DNA digesting enzyme, and we introduced that into this test tube. And when this was injected into the mice, mice was alive. It means that DNAs, that is DNA digesting enzyme, DNAs have digested the DNA present over here, and when this content was injected into the mice, this virulence property was not transferred into the mice, and mice was alive, and we can see no live strain, no live S strain. In this mice we got no live S strain. So this concluded that DNA is transferring the genetic material, or DNA is transferring the information from one generation to another, or DNA is the main heredity material. So with this experiment we concluded that DNA is the heredity material. Still there were some scientists or some biologists which were not, with those who were not convinced by this experiment. So later on Hershey and Chase, they perform an experiment which finally proved that DNA is the genetic material. So they proved that DNA is the genetic material, and they performed an experiment which is known as Alfred Hershey and Martha Chase experiment. So they worked with a virus, and that they worked with a virus which affect bacteria, and the virus which affect bacteria, that is known as bacteriophage. So here it's not written, but it's a bacteriophage. Okay, so the virus which affect the bacteria, that is known as bacteriophage. So they have performed the experiment with bacteriophage. Now what they have done, Hershey and Chase, what they have done? See, the bacteriophage attached to the bacteria. So when bacteriophage is attacking on a bacteria, whenever the bacteriophage attack to a bacteria, its genetic material, you can see here, its genetic material is transformed into, or its genetic material is given to the bacterial cell. So whenever the bacteriophage attached to the bacteria, its genetic material enter into the bacterial cell, and the bacterial cell treat the genetic material, that is the viral genetic material, as if its own, and then it makes its copy. So Hershey and Chase worked to discover whether it was a protein or it was a DNA which goes from a virus to bacteria. So what's the basic principle behind this? See, whenever a bacteriophage affect a bacterial cell, its genetic material or its substance, its, what we can say, genetic material, basically now we can say, so genetic material get transferred into bacterial cell, and bacterial cell consider the foreign genetic material as its own, and it forms its copy, or it replicates. So this Hershey and Chase, they wanted to know either this protein is going into the bacterial cell or the DNA is going into the bacterial cell. So what they have done? To find this? So they have done a very easy experiment. They have grown some viruses on a medium that contain radioactive phosphorus. So what they have done? They have taken some bacteriophage viruses, and they have grown them on phosphorus labeled DNA, or we can say a medium which was having a radioactive phosphorus. So it went into the DNA. So they grew some viruses on the medium that contained radioactive phosphorus, and they have grown some viruses on the medium that contain radioactive sulfur. So what they have done? They have taken two medium. In one medium, they have grown the viruses which were having. They have grown some viruses in one medium which was having radioactive phosphorus, and they have grown the other viruses, some of the viruses, in a medium which was having radioactive sulfur. So here we are having radioactive phosphorus in the medium, and here we are having radioactive sulfur. So some of the bacteria, they were having phosphorus, as we know that it is the main content of DNA. So some of the viruses, they were having DNA as radioactive. Some of the viruses, they were having protein as the radioactive substance. Why? Because sulfur is a part of protein. So in this experiment, what they observed, so again I will repeat, Hershey and Chase, what they have done, they grew some virus on medium that contain radioactive phosphorus and some other on the medium that contain radioactive sulfur. Now what viruses has done? Viruses grown in the presence of radioactive phosphorus, they were having radioactive DNA. But they were not having radioactive protein, because DNA contain phosphorus, but protein do not contain phosphorus. Similarly, the virus grown on the radioactive sulfur contain radioactive protein, but not radioactive DNA, because DNA does not contain sulfur. After that, radioactive phages were, now this radioactive phages, bacteriophages were allowed to attack on E. coli bacteria. Then after the infection, what happened? The viral coat were removed, and the bacteria by agitating them in the blender. So what we have done, when it has attacked to the, when this bacteriophage has attacked to the bacterial cell, then due to agitation or with centrifugation and all these things, the outer cover we obtained, the outer cover of bacteriophage, we obtained it in different in a different section, and we got to observe the E. coli cells. So we can just understand like protein or DNA what has transformed. Okay, so the virus particle were separated from the bacteria by spinning them into centrifuge. The remaining virus particles, so this you can see, this is the remaining virus particle. So this remaining virus particle were separated from the bacteria. This is the bacteria cell, and this is our bacterial cell, and this is the remaining virus particle. So this remaining virus particle was separated by the process of centrifugation, and bacteria which was infected with virus that had radioactive DNA. So this one. So the bacteria, so this bacteria, so bacteria which were infected, the bacteria which were infected with, so the bacteria which were infected with virus that had radioactive DNA were radioactive. So this bacteria, you can see, this bacteria which were affected by radioactive bacteriophages, they were having radioactive DNA. Indicating, now what it says, that when this bacteria have radioactive DNA, it says that, or it indicates that DNA was the material which passes from the virus to bacteria. So this experiment or this transformation tells that the DNA was the substance which passed from one bacteriophage to bacteria, and it was not the protein. And now the bacteria, now coming to this side, the bacteria that were infected with the virus that had radioactive protein were not radioactive. So here you can see the bacterial cell which was obtained, it is not radioactive. So this bacterial cell is not radioactive, and this bacterial cell is radioactive. Okay. So this bacterial cell is radioactive, and this bacterial cell is not radioactive. So this indicates that this is not radioactive. So this indicates that protein did not enter the bacteria from the virus. It states that the protein is not going to enter. This protein region is not going to enter into the bacterial cell. What is going to enter? DNA region. The DNA region is going to enter, but the protein region is not going to enter, because this is not radioactive, but this is radioactive. So this indicates that protein did not enter the bacteria from the virus. DNA is therefore considered as the genetic material. So in this, DNA is considered as the genetic material which passes from virus or bacteriophage to bacterial cell. So here you can see sulfur is detected in the supernatant. Supernatant means all the remains which were obtained after the centrifugation. And here And here you can see no phosphorus detected in supernatant. We didn't got any phosphorus in supernatant. It means that phosphorus entered into the cell. So phosphorus was detected into the cell, and here no sulfur detected into the cell. Here we got no sulfur, as sulfur was radioactive and it was in the protein. So protein was not inside the cell. So here we didn't got any sulfur. Okay, but sulfur was there in supernatant. Here no phosphorus was we didn't get any phosphorus in supernatant, and phosphorus was detected into the cell. So with this experiment, it was concluded that DNA is the genetic material. So finally, all the scientists, they believed that DNA is the genetic material, not the protein.